as you mentioned, the previous two lectures were mostly about the structure of the gene and the structure of the DNA and how the DNA is organized in the cell and also how the genes live in a population or the alleles and how the genotyping and imputation happens, and then asked me to spend some time on saying some different things about how to interpret the associations that you end up with after successfully performing a genome-wide association study or like a genome-wide meta-analysis of many different cohorts. So the main question one ends up is first if you do all the QC steps and all the other important steps, then you end up hopefully something that you can see on the left. So it's a Manhattan plot. On the x-axis you have the physical position for each of the S and P. Here we have, I think, roughly 9 million SNPs, and then on the y-axis you have the statistical significance from the linear regression, and everything that is pink, it's above the genome-wide significance threshold and what we consider to be like robust association. So it's called Manhattan plot, and actually, this plot was actually drawn in Manhattan, sitting in David's flat. So it's actually very, very nice, like many ways a Manhattan plot. But the problem with such a nice Manhattan plot, although it's pink and all, is still that it's very hard to say anything about the underlying biology, because if we go into this zoom in plot that you can see on the right, that is called locus zoom plot, and it's basically the same, where on the x-axis you have the physical position of this genome, and then on the y-axis you have, on the left you have the statistical significance, and on the right you have the probability for recombination, which is shown in the peaks. And then the content is, again, it's like all the SNPs and the statistical significance, but now they are colored based on the linkage disequilibrium between neighboring SNPs, and this coloring is done using the best SNP in the locus. So it's above genome-wide significance, but as you can see, all the different dots there that are also colored either by red or green or purple, they are have a considerable correlation between the leading SNP, and in a way it's almost impossible to say if the variant that ranks the highest in the region is indeed the causal SNP or anything else that is either in the perfect LD, meaning the correlation is 1, or very high, let's say like 0.9 or 0.8. And as you remember, or as I explained how the genotyping arrays were constructed, it can be that in this wide window, which is roughly 1 megabase, there are maybe only a couple of tens SNPs that are directly genotyped, and everything else is just imputed or predicted based on the reference genomes. And for this reason, actually, we don't know if probably this SNP that ranks the best is quite probably it's imputed, and it can be that why this SNP has lowest p-value is just because the imputation works slightly better than, for example, the other ones that are less significant but still in almost complete LD. And the things get, this is one of the reasons why it's complicated to say anything about the biology. Another thing is that if you look, down here, there are like almost, more than, 50 genes in the region that, could potentially be, linked or, causal or have an important role for the phenotype. And again, it's very hard to say like which gene in here it's the causal one. Of course, the easiest way is just to pick the one that is closest and say, oh, this is probably this ZPTB46. but most probably it's not. And this all like complicates it further. So, but what is the aim of this lecture is to help you to give some tricks how to define the causal variants or how to define causal genes, and then take a step further, like what are the pathways or biological processes involved, and also what are the tissues of action for our phenotype. So if you're thinking of like social phenotypes, and obvious the main tissue of action is brain, but if you have like a disease where it's not really clear what the physiology of a given disease, this kind of insights for the causal tissue could also be very, very useful. So, and also why it's important to find the causal variants is most immediate thing. It actually enables direct genetic testing for this different carrier status, or for example in pharmacogenetics, where there are different genetic variants that have an effect if a certain drug is going to work for you, or if what's the dosage you need to take to achieve the wanted effect, but also designing treatments or making treatment decisions. And of course it's also important to understanding the biology, developing new drugs, and gaining further insight. So it's in a way it's not that important for the questions that you have in mind, because you are more into this genetic predictors, having the score that you can use in your social studies, but it's more important from the biology side of view, where you really want to understand the genes. Yeah, Dan.
I actually think that there are uses in social science where it is important to know what's causal. Like we're going to talk about Mendelian randomization studies where you have to make an assumption called the exclusion restriction, which in order to know whether that's likely to hold, you need to be pretty confident about what the biological mechanism is for the gene. And also, if you want to use a genetic variant as a measure of a structural parameter in a model, you're making an assumption about what it's actually doing biologically. So, I think what you're talking about is not only important for medicine, but also
yeah, definitely. And it's also like, especially in the Mendelian randomization, where you actually have to know the path, which way the variant has its effects on the end phenotype. You really have to know that you're excluding all the pleiotropic variants that have some other paths to your end phenotype. So yeah, indeed, it is important to causality in many ways. But in basically, what I wanted to say is that if you are constructing a score, then in score it's not that important. But of course, in some analysis, indeed, it's important to know the causal variant, and of course for the score as well. If you have the causal stuff in it, your scores are much more precise and more predictive. So I think the limitations of GWAS we already talked, that we are, as the genome is structured as haplotypes or long blocks, and we are still just looking at the association with the haplotype or for a block. And that's why if we draw this locus zoom plot, we actually don't really see like a single signal, but we usually see like a plateau within this haplotype, where on the edges the significance fades away, but it's a pyramid or a plateau. But it's and within this plateau, it's quite difficult to say which of the variants is causal, or even if the causal variant is captured, because it can be a rare variant that is just within the common haplotypes. So there are many ways how you could find the causal variants, and here I will just walk through. And one thing is that if you like look into within your loci, you want to identify if there are multiple independent signals, and try to define this independence. Traditionally the best way is just to take your your lead SNP in the loci, locus, and just include into your linear regression model. So basically account for this genotype and see what is left, and if there are more SNPs still genome-wide significance, and then you can do it, over many iterations. And there are now methods that you can actually, you don't really need to have the raw data yourself, but you can just work with the summary level statistics already, the output from your genome-wide associations, and then use some reference LD matrix, and you can then define the independence. And there is just like a couple of, one example from our, this recent high paper. I was very closely involved, and there, as I mentioned, we had almost 700 genome-wide significant signals defined as, the distance between the different SNPs were at least I think half a megabase. But then if we do this multiple, regression, trying to find independent signals, we actually found that there are, roughly like 1/4 of our loci actually have several signals within it, and there are in total like 260 or more secondary signals in your loci. And this is just like one of the examples below, again like locus zoom plot, and here the causal gene we know is agricon. This is the gene that, for example, for Danny DeVito, is mutated, so his long bones are short, but the other proportions are okay, but just the long bones are short, so he has a mutation there. And here, for example, all the different colored dots are the independent signals that we came up with. And so, for example, like within this agricon gene, there are 4 different independent signals. So it's clearly like, all causal, because it's like several signals point to the same gene. But there are, as you see, like there are 2 other variants here, and those are, this gene independently has also been linked to like this familiar short stature, and also here there are some evidence, and here those blue peaks are like this recombination hotspots. So basically it means that, here we have a independent haplotype, and here we have an independent haplotype, and those 4 variants also, those are within one haplotype, but there is another haplotype. So it in a way shows that also the biologically similar functions, or genes with similar functions, cluster same. But also another thing how you can, guess that, a variant could be, causal, if you try to see if there are some like non-synonymous variants. So the SNPs that change the codon structure, so it changes which amino acid gets integrated into the protein. And quite surprisingly, those 4 variants within this agricon gene actually independently tag 3 missense variants. So although those missense variants were not directly available in our panel, we could infer that they were independently very strongly correlated with the non-synonymous variants. So changing the protein structure or its functions, and this way we can actually say that probably those non-coding variant, or those non-synonymous variants are coding. So there are some computational tools where you can find more signals. Then another very good tool to find causal variants is actually to look for the gene expression. I already explained in the previous session that how the gene expression is basically the measure of if a gene is active or not, and then we can basically check if the variants or the SNPs that we identify in our genome-wide association study are those specifically affecting the gene expression on any genes like within close vicinity. We usually consider like plus minus 1 million base pairs as the the window And as I said, like it's very simple. It's just, you take the SNP genotypes and then run a simple linear regression to see if the allele dosage has a linear effect on gene expression. And when we, repeated this, or we did this experiment with the HapMap2 panel, so the 2.5 million SNPs, then, surprisingly, out of those 2.5 million SNPs, 20% actually had a statistically significant effects on gene expression in blood. And this is, it's like 20%, it's roughly 500,000 SNPs. And statistically significance, I don't mean here like, the nominal significance, but really, significant after running 100 permutations and that false discovery rate of 5%. So this is actually, very important result to remember, because if you just take your your GWAS signal and just check if in some of the database your SNP has an effect on gene expression, it doesn't mean that this gene is causal, because this gene may, or this variant could be in very, very strong or strong LD with the causal variant for this affects the gene expression, and what you observe is just a residual effect on the gene expression. And again, quite surprisingly, roughly half of the genes in blood, we could detect like a variant, genetic variant that affects the gene expression. So in a way, it proves that blood is a rather good model for finding the causal genes, or the variants that we identified in our analysis could have some functional consequences. Some characteristics about this EQTLs, or expression quantitative trait loci. Here, the first plot up in the right. Again, here we have on the x-axis we have the distance between the the variant and the gene, and then on the y-axis we have basically a measure of effect size in the set scores, and you can see that actually the strongest effects are within 50 kb from between the SNP and the gene, but still there are rather strong effects detectable still that are as far as 250 kb away. And here, if this is again on on the second plot on the up, it's like again the distance between the gene and your SNP, and then on the y-axis you have the fraction of all the cis EQTL SNPs within this window length. Here again you see that a bulk of the important SNPs are within plus minus 50 kb, so indeed very close to the gene, but at the same time there are very strong cis QTLs that are very far away, and 95% of the EQTLs are within plus minus 250 kb. But if you want to get 99% of all your significant EQTLs, they are as far as plus minus one megabase away. Another very important thing to remember is, as I mentioned in the previous graph, for the height, that there can be like many variants within the gene that are affecting your height. Here what we did, we tried to estimate like how many independent SNPs, so independent meaning that they are in different haploblocks or in different haploblocks or haplotypes. There are most of the genes still have one significant regulator, but there are again sizable amount of genes where you actually have more than 6 or and up to 16 different regulatory regions or regulatory blocks that we could identify. So it's if you are measuring a phenotype that is biologically so close to your genotype, And with that I mean that if you have a, for example, a SNP within this regulatory region binding site, it has a direct effect on how much gene expression there will, or how abundant the gene will be expressed. But in education, it's so distant from your SNP that there are so many mechanisms in between. So you need, in order to, for education, we find 16 independent signals for a given gene in a locus. We really need very, very large sample sizes. And here, the last plot, this is where we have a variation explained by a single SNP and a variation explained by combining all those different SNPs per gene. There you can see that first that actually the effect sizes that we are talking about are much larger. For example, there are SNPs that explain up to half of the variation in the gene expression, but if we combine those SNPs together, you get a better score, can get up to 70%. But again, it's the first SNP usually explains the most, and then it's flattens out. And in this study we actually had so much power that we could ask, because we could find the cis-equity effect for every gene, but we didn't find. And then we asked the question why we didn't see the significant equity effect on some of the genes. We just took all the genes together, run a pathway enrichment analysis, so basically estimating what pathways or what biological processes are represented in the set of genes. And actually it turns out that those are mostly the genes that actually are important in this basic cell maintenance, like all the processes I explained you previously, that they don't really tolerate any change or this slight modification. So mostly the genes that can be affected are not so important for the spatial functions. And again, if we discuss a little bit this distant effects, and what we did here, we just took all the Genome-Wide association signals by the time, so it was 4,000, and across a wide range of phenotypes. And then we again asked if those SNPs have a direct effect on gene expression across all the chromosomes, and quite surprisingly, 8% of all out of the 4,000 SNPs actually had effect on gene expression, regardless of the chromosome. And this is just what you see on the bottom right is a locus zoom plot. Oh no, it's a circus plot where you, and then in the circle there are the SNPs, and outside are the gene names. You can see there are some like hotspots that are more important, like globally regulating, and using this type of analysis you can actually infer this more global effects of your variants of interest on gene expression, or then on pathways. So it's not, again, maybe something that you can implement directly yourself, but it's more to so that you can understand that the variant that you identify in your GWAS may have much wider effects on the biology than just the gene nearby. And something that very, very important for the the Mendelian randomization and identifying the causal variants is that in our case there are there are some variants that are linked to many different diseases. So it's hard to say. You cannot really use this type of genetic variants in your Mendelian randomization studies, because you don't really understand clearly what is the mechanism, the biological mechanism, what this SNP is having this effect through. And it's the same for us, that, for example, we we observed that there are some SNPs that have effect on wide range of different genes, and those genes are actually overlapping between quite distant phenotypes. So again, I say that the medial randomization is very good approach, but you have to be very careful that you know the biology behind the question before you implement your statistical frameworks. Again, this we already discussed, so let's skip it. So so this is like the ways how to find the right gene. You either like try to find variants that are in strong linkage equilibrium, that are non-synonymous, so maybe like affect the protein structure, or then you use gene expression to estimate if the variant you have identified have a direct functional consequences. So now if we take like a step, I don't know, further or back, and can we use some methods to identify the right tissue, and now there are many tools available, but one that I highlight here and I found it really useful. It's called Depict, and it's in principle what this tool does. It actually has gathered publicly available gene expression data for 80,000 different arrays, and this is both the humans and animals. And what it has done is like run principal component analysis across this matrix of 80,000 arrays and 20,000 genes expressions, and then try to identify the correlations or the loadings on those principal components across different genes. And then what it enables, because for some of the genes we already know the functions across from the either like animal models or molecular biology experiments, but for some we don't have any clues whatsoever. And then using those loadings on the principal components and knowing the function of some of the genes, we actually can predict the function for other genes. So in a way we can go beyond the known biology. And this tool enables us to prioritize prioritize pathways, and also prioritize tissues, because we can predict the functions, and we can also predict where a gene is expressed where there is no existing information in the databases. And how it works is like we, for example, here in the top, this is like our familiar Manhattan plot, and what we can do, we can just take all those regions that are genome-wide significant in our study, and then we across all those in this case 3 regions, we take all the genes within a certain region, and then we look up all the predicted functions for those genes, and then we can even though there are like genes that are not causal for our phenotype, there are still more genes that are causal, and then in this way we can quantify if there are any pathways that come out. And here we basically see that lipid metabolism seems to be one common denominator for many of the genes, and so we say lipid metabolism is probably one of the pathways where our genes are active. So, and now when we have identified the pathways, then we can again look across our loci and see which genes fall within the predicted significant pathways, and then we can identify genes. Or we can do the same thing, look the genes that are fall within a given biological pathway and see where in which tissues they are mostly expressed. And this is this third panel here. You can see again that liver, those genes that we have identified in our loci seems to be definitely overexpressed in liver, and knowing that lipid metabolism acts in liver, so it's a very powerful tool. So yeah, I really recommend to use it to to prioritizing genes, biological pathways, but also tissues. And this is like one real life example from the very recent educational attainment paper, where we had I think this 165 loci, but I think those analyses were run with the initial 779. So basically we did the same thing. We took all those 79 regions in the genome and then took all the genes within those regions and then run those gene expression based expression methods. And then in the left, what we did, we just estimated in which tissues the genes that fall within our loci, they are like more expressed more than one would observe by chance. And and we used here actually a little bit different gene expression database. It's called GTEx, and there they have more than 10 different tissues from brain available, and it's pretty clear that the genes within the education attainment loci are more expressed in brain than any other tissue. And this is one way how we can basically use the summary level data to learn something more about the biology, in this case identify the causal tissue. And this is just another example, again using gene expression and public available data of gene expression profiling across different developmental stages. Like it's like the prenatal, so before the birth, and then postnatal, where you have like the early infancy and childhood and adulthood. And then again we can take all the genes or loci and ask are they expressed in brain. Indeed they are more expressed, definitely more in brain, which is the red line compared to the black line. But then also we can ask like if in which part of the development the genes play a stronger role, what we identified. And if we run like a direct t-test, we can actually say that yeah, the genes are more active, or at least are more abundantly expressed in like prenatally than postnatally. So again, something can be, now if you have already identified the tissue of interest, then you can already look into more detailed question to again learn something more about the biology. Yes?
So is the idea that the SNPs themselves are doing something that directly affects the gene expression, or they're just associated with some other thing that we don't know that is affecting?
I think in this case, as we are not like really using the SNP effect on gene expression, so we are not assuming that the SNP has direct effect on the gene expression. But as we are using basically here we are using the SNP as a beacon to define the region where we are looking into more specifically. So yeah, we didn't check if any of our, actually we did check if any of our educational attainment SNP actually also affected gene expression in brain directly. And yeah, indeed we identified I think around 20 genes, but not that many, again because it's having blood tissue it's very simple, but having brain tissue it's rather complicated, and there are not very big numbers, and usually if there are some brain tissues available, they are not really healthy donors, but they are like ill for some diseases, or even if they are healthy donors, and if it's not just accident, there can be some other modulators. So yeah, we are not actually assuming here that the SNPs have directly effect on the gene expression, or they may have, but we don't have the power to test it. But it's more like to have a prior to identify genes that are most probably the causal candidates, and then we can, having this knowledge, we can then ask like, are they indeed more expressed? And then, yep.
And so what would be like a plausible biological mechanism? Like what would have to be going on in that region? If the SNP is just like a signal.
No, it's not signal. It's like the SNP doesn't need to be the causal SNP. So this is, I think, what I'm like to say. Probably they're still there in the DNA. There are the regulators that are regulating the gene expression or some response to the environmental changes. But I think in this case, what we really wanted to improve over our previous paper, where we identified the 3 SNPs, where we didn't really find that good biological insights, but here to show that that basically it's like a proof of principle that in education, as our phenotype, we identify that indeed we have overexpressed expression in brain. And if contrast, I don't have here a plot, but if I would take my 600 height associated variants and run exactly the same exercise, then I would end up with bone and cartilage as are my main enrichment things. There will be like others as well, because like height is like all development, but those were like like highly significant. So it's in a way it shows that the tissues going along with your phenotype. Yep. There was other question? Yeah.
Yeah, so I thought I could maybe come up with a hypothetical example that might help. So one of our 74 SNPs is a coding SNP. It's located in a gene called leucine-rich repeat in neuron. Presumably that SNP, its causal pathway toward years of education is not through affecting gene expression. It directly affects the structure of the protein encoded by this gene. But nevertheless, you would expect that there are other regions that regulate the expression of this gene and to make sure it's turned on in the right tissues at the right time. And therefore, that gene would still contribute to all these patterns that we're seeing in these plots, despite the fact that it is not a regulatory SNP, it is actually a coding SNP. I mean, does that help?
Yep.
So this may be a very naive biology question, but I know that allele frequency varies by population stratification. Does the correlation or association between alleles and genes or genes and tissues vary by population stratification? I would assume the latter would be no, and the former I just have no idea.
So the power to detect if a SNP is affecting gene expression again depends on the allele frequency. The rarer the allele, the lower is your statistical power to find this effect. But now your question, like, is the gene expression systematically different between different ethnic groups?
I mean, it could be.
There may be like some, for example,
there was an EQTL that was not the real causal SNP, it was just high LD with the real EQTL. Conceivably, in one population, you would see that SNP is strongly associated with expression of a particular gene in a particular tissue or whatnot. But then you move to the next population, LD differs by population, that SNP is no longer in strong LD with the true EQTL, and it would no longer be an apparent EQTL in that population.
Yep. And there also can be like, in different populations, indeed, some of the genes may have, different expression levels in the same tissue. For example, if, are living in the high mountains or in the lowlands, there may be different requirements for your lungs, and that's why the gene expression level in the same tissue can be differing just because of the environment. And the same is for immune system or any other thing. So but here again, it's the population stratification. This don't really matter, because we are just using our association, and then we are just picking the genes that we are having a more closer look. But this is like using the gene expression, but we can ask the same question, like, can we identify relevant tissues to actually using those the epigenetic marks, what I again briefly touched based on my previous lecture. Like, there are a set of different epigenetic marks which are specific for different tissues. And then again, here we can apply some methods that Patrick will dive into later on this course. But basically, we can quantify how much genetic variance can explained across those different tissues, having information on those epigenetic marks. And here, again, we already included, the years is in blue, but there we also have as like a nulls for like body mass index, waist pressure, and height. And here, you can see that, for height, if you use the height SNPs, most of the genetic variance gets explained in regions where, there are epigenetic marks in connective tissue or bone that sign that the DNA is active, and the lowest for height what we have is like immune system or central nervous system. So there is a significant change. But if you like repeat the same experiment with education attainment, there we actually see that the strongest or the most genetic variation gets explained in the regions that are active in central nervous system, and this again provides like independent evidence that the brain or central nervous system indeed is the tissue of action, because the genes there are more expressed, and also using those different epigenetic or molecular marks that show that the DNA is active. It's also like the most active in the brain. So this is like those more comprehensive summary level approaches that allow us to identify the tissue of action. And again, I think the example of human height, it's really standing out that this indeed explains the most. Of course, like the error bars are quite wide, and so, but it's still indicative somewhat. Yes.
Is it surprising at all that the CNS region is not actually super enriched for any one phenotype, even though you might expect it would be way more enriched for edge-of-years than the other things?
Yeah, I think there are like two or several factors. One is that this the variance explained with our Genome-Wide association data is like almost 10 times less than for height. So you're actually not capturing that much of the variance or that much of the biology. So that's why you don't see like a boost, and there is like this background explained as well. And the other thing is like probably the bone tissue is much more well understood, again because of the availability of the biological sample. So you could go for profiling for the epigenetic marks or DNA accessibility marks compared to brain, which is again like not so abundantly available. So there are several functions. Yes, James?
Yeah, and I would also say that different categories have a higher ceiling for enrichment than others. And CNS has a pretty low ceiling, because actually a pretty large percentage of all SNPs are in regions of the genome that are wound around histones that are observed to be marked in the central nervous system. So one suggestion I can make is actually to read our online supplement and be really dedicated. And in our section about stratified LD-square regression, we provide an explanation of how to interpret these enrichments and the output of this particular analysis that'll probably make more intuitive sense. If you want, years of education to be really in the brain and height to be really in bones and cartilage.
And also, like another point of thing to point out is that actually half of the genes or so are like expressed in brain. So it's quite tricky to say like if the enrichment is abundant. At the same time, this also provides us more power to see the enrichment if it's indeed it's expressed in brain and it's not really expressed in anywhere else. So, but it's just like an example how we can use different biological information and summary level data and clever statistical approaches to infer something about the underlying biology. So finding the right genes, I already talked about using the gene expression or eQTLs or the non-synonymous variants, but there are some other tricks as well. One of the tricks is actually looking for like known biology or known associations. And as I mentioned for the very early on about this height association with aggregate, so it's a well-known Mendelian condition, and quite surprisingly in a way we find like both like Mendelian mutations and common alleles having effect on the same gene. But again, it's the same analog that it's different thing if you're driving a car and you are missing a wheel, or just the pressure is different. So here we actually took the same approach. Together with James, we identified like a list of genes noted to be linked to various cognitive diseases like schizophrenia or intellectual disability or autism spectrum disorder, and we also looked actually for microcephaly. And then we just more broadly just took the genes within our loci and then run the depict on it, so which prioritized us a set of genes. And then we took those genes and looked the overlap with the genes that were linked to different conditions and have like de novo mutations, which means that they're not present in the parents but just occur. And quite again not surprisingly, but we observed like very strong overlap with all those different conditions. And again we run some enrichment analysis again, like how much we would expect. And here again we have like the education attainment in blue, and also height, BMI, and waist-hip ratio as nulls. And here you can see that for all those three categories, definitely we see a huge enrichment for intellectual disability, autism spectrum disorders, or schizophrenia. But not that surprisingly at all, there is not strong enrichment for abnormal skeletal growth genes. And if you look for height and BMI, there is like much stronger enrichment there. So it's one way how to prioritize genes is like using this kind of summary level data, and basically let the data to tell you what genes are probably causal, but you can also like use the existing knowledge to what is linked to the Mendelian diseases, and then use those as a way how to prioritize genes that are probably causal. Again, this is just like another example. What you can, if you're increasing the sample size, again now I'm switching back to the height, previous GWAS, but there is also like tools, as I mentioned, like you can use gene expression to prioritize genes, but there are also tools that actually use abstracts, like PubMed abstracts, so they do like literature search, and it's called like a Grail. It used to be a very popular tool in the GWAS field for a while, but now I think it's pushed out by the PECT. But basically what Grail does, it looks for if those two genes or your RS number and the genes are co-mentioned in a PubMed abstract, and if they are mentioned together in the abstract, then it prioritize those variants and genes. And this is like just one of the representation of the things, like this interconnectivity, and where you have only 100, like say 200 loci to consider, and here you have 700. This kind of again summary level search over, for this case, like literature really provides you a lot of new insights, or helps you to prioritize genes. and there are also available like pathway prioritization tools that you can also take in the your genes or your loci and then try to estimate what are the biological functions captured by your loci. I think this is this is again like a quick snapshot to our Nature paper on educational attainment, where we try to summarize the top genes that we think are causal, and this is in a way summary of all those different approaches we took. This is like what is the the loci in the gene, and is it like in identified in like intellectual disability or autism or schizophrenia, and then it's like this depict prioritization p-value, and but then also across a wide range of tests, like is this gene is there a variant in like non-coding, like non-synonymous? Is it like EQTL? Is it expressed in brain? Is it implicated some other ways? And there is just like the pathways that are highlighted, and as James keeps telling, those results just explain the full path from birth of a neuronal cell to the really fine-tuned information flow. So this is really like insightful, and there are some things that you can really like learn. It's quite surprisingly like genes that are linked to Huntington's disease or Alzheimer's really also pop out as in the loci that we have identified for educational attainment. And there are like other insights from really like biological processes that also are biologically logical, as I like to say. Like for example here, like axon genesis, it's like how the axon grows, or how the different information flow through the different potassium channels or different signaling. So it's again I think the best way is really the best tool to use is the PICT, because it provides you so many insights to the biology. And although it's a little bit like a black box, you put things in, you get nice things out, but as long as the nice things make sense, I think it's justified. And I think I ended here like with again this kind of looking for the pathways the same way. We identify the use the SNPs as beacons and identify the genes, and then look in which pathways they are fall mostly in, and then we can identify the pathways that are abundant. And then here we can also look like how the different pathways are interconnected, and we can learn something more about the biology, or in this case just reinforce the notion that the things that the GWAS that we have conducted is not just is not rubbish, but provides some meaningful insights. And this is just like a couple of the tools and names that I recommend to use, and if there is of like interest to have like a little bit more extensive understanding. And then I think as James pointed out, I think the supplementary methods for the EA2 actually provides a very good overview of the state-of-the-art tools and approaches, and it also provides a very nice example or explanation why those tools are good and what you can learn from this. So if you have questions, I'm happy to answer, or either for this last presentation, which maybe got like too much into the things you don't know much about, but it was a little bit future looking if you are starting to conduct those studies, and also in any of the other topics I discussed before. So please. Yes, please.
In the subjective well-being paper, it talked about inversion polymorphism several times. Could you explain what that is?
So I didn't. Is there a whiteboard? Anyway.
Pull up the screen.
But maybe I can explain. So basically, it's an inversion. It's let's draw old school. So basically, if we have let me draw here a chromosome. So this is the chromosome. And here, for example, you have a, let's like say this region, and then what is an inversion is that it's like A and B, the breakpoint. So in a normal chromosome, you have like the A is closer to the end than the B, but when there is an inversion happens, then the chromosome looks like that. So the B is here and the A is here. So it's inversion. It means like it gets basically cut out, and just the orientation gets reversed.
And how can you tell that apart from just a snip?
Um,
Is it because the whole thing is flipped around?
So basically, because such things happen, the linkage, basically the recombination gets disturbed in those regions, and actually I guess in subjective well-being, I think one of the inversions was again I don't remember how long was the 17 chromosome one. No. It's like many millions of base pairs, and this actually disturbs the this crossing over event. So actually within this block in the set of individuals who are carrying this inversion, there is a very long range LD. And how we actually detected this was that we did like a locus zoom plot, as I showed, and then you have here the oscillation, and there is just like a wide region where you had a very significant block, and this, as we said, is like many megabases long. So this is one of the indications. And then what we did, and this is how you can analyze the inversions. You actually focus on this region between the A and B, and then just run a principal component analysis. And and then you run some, and this principal component analysis actually enables you to, based on the loadings on the first component, you can actually, classify the individuals either carrying the inversion or not. So this is, I think it was a very interesting finding to me, especially because there are not that many inversions linked to a phenotype. It's I think the chromosome 17 is linked to like fertility, but not really like this kind of social phenotype or like medically relevant phenotype. And it was the chromosome 17 we had such a inversion event, but then also in chromosome 8. And I think in the next round I'm very eager to do like a systematic search if there are other inversions. And now if you ask like what's the biological relevance, or can we like have a really huge effect, completely messes up because the gene orientations and stuff get mixed. Actually, all the genes that are in the middle, those are completely okay there, because all the regulatory variants that I explained are still close where they are. But now for the genes that are close to the breakpoints, for example, you have a gene here, let's say, but all those the regulatory regions are here now, because it used to be like the vice versa. Then it's actually the expression of the genes that were close to the breakpoints were actually hugely or very strongly affected by the inversion phenotype. So yeah, it's just like another. I discussed this tandem repeats or copy number changes, but it's just another form of genetic variation that is not usually captured. So, more questions? Yes.
So going off of that, are there any other structural changes that you can pick up with SNP chips? Because I know there are like just duplications. Maybe the genes like transpose themselves, just like deletions of large sections sometimes. Like, do you ever observe that?
You can. You can, using SNP arrays, you can identify, inversions in certain extent. You can identify the copy number changes, like the duplications or deletions, especially if they are very, very large. What you can even do is you can identify if duplication or deletion is only present in a very small fraction of your cells. For example, in blood, let's say the duplication is only present in 5% of your cells. And you can even use this arrays to actually identify those. And again, how you can do it, because you have the allele frequency that you can estimate, but then you also have the intensity of the signal. And if it's not just like red, yellow, blue, but then the yellow gets to be like reddish or greenish, then you can actually quantify the how it's distributed. So you can
You can identify aside from like maybe a quality control issue or anything like that.
Yeah. Because again, it's not really quality control, because it usually this kind of very long changes happen, like let's say like half a chromosome or like a big chunk. So it's not just like a single SNP that you use, but it's like a systematic deviation or a systematic pattern. So yeah, the inversions, copy number changes are the main things. Those so those tandem repeats, if they are linked with the SNPs, you then can impute those, but usually not. So there are not that many things that you can infer from the array data, but still, yeah, you can. Basically, this way it's also very cost effective to do the arrays, because you can also infer the copy numbers, and the copy number changes actually have a effect on cognitive abilities, but those are not very common, but more rather like a rare end mutations. More questions? Everything's clear, it seems. But I will be around until Sunday, so if you have any impromptu questions, or I'm happy to explain in more detail, or provide the links. I already pointed out that the materials that are under my name, that they were like optional, so there is no need to read them. But there are several very good reviews on, for example, the imputation and the array design, or this linkage equilibrium and the use in genetic studies. So if some of the topics that I discussed today seemed interesting, or you want to really go into the math and understand how the imputation works, so this is what you can find from those papers. Yes?
I just wanted to ask a follow-up question to my previous one, which was about, if I understood you correctly, you said that the gene frequency in tissues can vary, right?
I said like the gene expression levels.
The gene expression
Yeah.
levels, right? So the frequency or the level of gene expression can vary within a tissue. Can the genes in a tissue vary? Right? So is there some set of genes that are always associated with some tissue?
Some genes are only expressed, let's say, in brain and never anywhere else. And then there are a set of genes that either are actually expressed in every tissue, regardless of the even like the time point. So they are like constantly needed, but there are some genes that only needed in certain tissues at a certain time.
Okay, and so while those levels of those specific tissue genes, whatever you want to call them, right?
Yep.
While those can vary, they do not vary across population stratification.
Again, I think this is not really systematically looked into, but we can assume that they are not, if the conditions, environmental conditions where the populations are living are the same. But as I pointed out, for example, if you are looking for people who live in the in the you know in the mountains, or they are living close to the coastline. There the environment can trigger some changes. And again, if you then running like an EQTL analysis, having those two populations, it's not then just genetic stratification, but then there can also be like environmental stratification. So it's like it's a double complicated problem to get rid of. And that's also why when you're doing the problem sets and you have to include those principal components into your analysis to avoid the stratification. This is indeed because, as I showed, if you just use like the SNPs and run the principal component analysis on your study populations, you can see that the people in north are systematically different than people in the south. And if you are not, it doesn't really matter what's your phenotype, but if you are pooling your data from North and South Europe, and you're not controlling for the place of birth, you end up with association with lactose intolerant loss. So it doesn't matter what you are studying. Is it like education or subjective well-being or religious orientation? You always end up with lactose intolerance loss. So that's why it's important to control for stratification. I don't know if I now answered your question. Okay. More? Yes?
So when you get into the discussion of matching, I guess linking that with descriptive data on expression, that's all from databases that have just been put together on which genes are co-expressed. So it's sort of like, is that co-expression used to sort of infer how the GWAS genes are interacting with each other, or it's just a separate data set that's being brought in?
Yeah, it's a completely like separate database, and it's just like the like a raw gene expression from all different experiments, and then within this data set, the co-expression patterns are estimated. So it's like completely independent on your GWAS, but you just use this co-expression matrix to first learn, go beyond the known biology, because you don't have to have like already experimental proof that there's a gene is active in certain way or certain tissue, because of this, knowing some genes have been annotated and others not. So yeah, it's not linked to your GWAS in any ways.
Is that how you inferred like the trans EQTL stuff?
No, in trans, there we really had like the individuals and the gene expression in our hand. So we had the raw data, but basically inferring the functions for the genes that come up. We don't have to work with our own data. We can use like a database in principle to predict the functions for those genes. Okay, but again, try to explain later if it still fades the understanding. Okay, so I think I've fulfilled my 210 minutes. So I hope you learned something, and if not, just feel free to ask me questions. Okay, thanks.